I'd like to welcome you to this presentation, Pathways to Success, Collaborative Cancer Research and Therapeutic Development at Cornell. And my name is Lauren Warnick. I have the pleasure of serving as Dean of the College of Veterinary Medicine here on campus. We have at Cornell a long history of comparative research benefiting both animals and people. And I want to welcome all the members of the Trustees Council, family and friends to this session. We'll, after the presentation, we'll have a few minutes for questions, so listen carefully. That'll be you asking the speakers questions. We won't quiz you on anything, so, so listen carefully, look forward to your questions. And our two faculty speakers are, first of all, Dr. Lou Cantley from Weill Cornell Medicine, and Dr. Kristy Richards, who has a joint appointment in the College of Veterinary Medicine here on campus and at Weill Cornell Medicine. So with that, I'd like to welcome Lou Cantley, and I think you'll take the lead.
Thank you.
Thanks.
Okay, let me know if you can't hear me back there. Just raise your hands and wave, and I'll try to speak up. So I was a Cornell Ph.D. student in biophysical chemistry, Baker Labs. Got my Ph.D. in 1975. And if you told me at that time that later in my life I'd become the director of a cancer center, I would have not believed it at all, because I'd actually never had a biology course. And you might have thought that would be required. It turns out it's not. Instead, I worked on enzymes and metabolism as a graduate student, but ultimately made a discovery in the mid-'80s or late '80s that revealed a gene that we now know is the most frequently mutated gene in all of cancer. It's also a gene that mediates virtually everything insulin does. And that's actually linked insulin resistance, and cancer into very surprising connections that we're still trying to understand in detail. But I'm not here to talk about my research. I really want to talk about how, at this stage in my career, as I was asked 4 years ago to take on the directorship of the cancer center, to me, this was a tremendous opportunity, because we're looking at a revolution in cancer right now, in our understanding of cancer and in new therapies. There's no other disease that we're making as much progress as we are in cancer now. Targeted therapy, immune therapy, and it all comes down to understanding the science behind it. When Nixon declared war on cancer in 1971, I think it was, we had no idea what cancer was. We hadn't the slightest idea what it was. We just knew it was hypergrowth, but we didn't know why. We didn't know whether it's caused by viruses or metabolism changes. It was just a mystery disease. And we didn't make much progress. But now we're beginning to understand what it is, and we are making progress. You have to know your enemy if you're going to beat them. But it's also clear that we have a long way to go. And the problem is to figure out what patients should get what drugs, or what are the new targets we should develop drugs against, and particularly how to use drugs in combination. So how do we set up an infrastructure so that we can take the basic science discoveries made in the laboratory and convert them into new therapies in a very short period of time? Typically from the time you find a potential drug target, many of you have heard of HER2 and Herceptin, breast cancer treatment, the HER2 positive breast cancer. Well, from the time HER2 was discovered in 1984 until the drug was approved around 2000 is too long. That's 16 years or more. And that's typical. That's been typical, to go from discovering what's causing a cancer to getting a way to treat it. 16, 20 years is not unusual. So how can we speed that up? That's what I'm going to talk about, the opportunities of coming here. I realized that Weill Cornell was just about the right size without people siloed. That you could get the basic scientists, the clinicians, the surgeons, all to work together in the same room, teach each other the same language so you can talk to each other, and figure out how to convert an observation into a new therapy. And it turns out the vet school, and particularly our hiring of Kristy Richards to be our emissary between the two, and I think many of you know, Kristy is an MD Ph.D. from Stanford, actually an undergraduate from Cornell, much later than after I left. And so she can speak in both terms. She knows the basic science, she knows the clinical, she sees patients with lymphoma, but importantly, she also sees dog patients with lymphoma. And it's a huge opportunity to be able to learn from treating dogs with the same drugs that we're treating the patients as to who responds and who doesn't, because dogs are much more inbred, and it's easier to figure out the genetics of response in dogs than it is in humans. And so, but Kristy, I'll let Kristy talk about all that. I just want to tell you a bit about the infrastructure we've set up just in the last 4 years since I arrived at Weill Cornell. So what's front and center, and the main thing that I was really interested in starting when we began the Cancer Center, is something called precision medicine. And basically what precision medicine means, that you look at individual patients, look at their tumors or whatever disease it is. It doesn't have to be cancer. Precision medicine is more broad than cancer. But in the context of cancer, where precision medicine is having the biggest impact, what it really means is when you take that biopsy or surgical material, you dive deeply into understanding what's gone on in that tumor at the genetic level. What mutations have occurred? What oncogenes have changed? Now we're even asking what immune cells are there. Why isn't your immune system killing the cancer? So there are all kinds of questions we can address by deeply diving into what's going on in that tumor that were not even possible to ask even 10 years ago. And so this is, I asked Laurie Glimsher at that time, who recruited me here. The two of us asked Mark Rubin, a real world's expert in this precision medicine technology, to head our precision medicine team. And what Mark has managed to do in a remarkably short period of time is get approved a New York State approved CLIA compliant assay that allows us to sequence every single gene in that cancer. Full sequence, there are roughly 25,000 genes. We now sequence all 25 in the patient's tumor, and it's done in a CLIA compliant, which means that based on what we see there, we can make a clinical decision. So if Kristy gets a report from one of her patients, almost any gene that's mutated, she can use that information to decide on a therapy. And we were the first institution ever to get that done in New York. I think the first in the world. And we went from really not doing much at all in this area to really being at the very front. Now we also do something else called RNA-seq, which means we also sequence all the messages from the DNA that tells us not only what aberrations are in the DNA, but then how does that affect expression and proteins that are being made by the DNA. So we had a tremendous amount of information that was never available before on every single cancer patient. And the consequence of that is that in most cancer patients, while in the past, even 5 years ago, only about 10% of them could we find anything that explained why they had the cancer. Now it's something like 85% of the cases. We can look at this and say, we know why that person had the cancer, or even why that cancer metastasized to the bone or to the liver or to the brain. And that's just revolutionary. Now, translating that knowledge into actual therapy is the next problem. And that's what we'll be talking about in the next few minutes. Now, because we'd set this system up, we were recognized by the US government as one of the forefront places to do this kind of research. So there was a referendum that went off to ask various institutions to apply to receive roughly $40 million in order to contribute to sequencing the full genomes of ultimately 1 million people across the United States and across various ethnicities, so that we could understand the variation in sequences of genes of people and their likelihood of getting a disease. And so we managed to be only one of 4 institutions in the country to get this award. We actually shared it with Columbia, in New York Presbyterian, but our site at Weill Cornell got 20 million of that, and we're responsible for sequencing 150,000 patients. But more importantly, we're going to be following those. They aren't necessarily patients, they're just people that we see who come into our hospital for their yearly checkup. So every year we will take blood, and over a 10-year period of time, we'll have blood from these individuals. We'll get their full genome sequence, and we can also interrogate in the blood and ask, can we see little fragments of DNA or even circulating cells from tumors that may have told us long before the patient even has a cancer that there was something brewing there. And so we have a tremendous opportunity to learn how a tumor evolves and what we can use to detect it much earlier than just an image where the tumor has to be pretty big before you can see it. Turns out we can see incredibly small amounts of DNA circulating, and so we're very excited that this is going to be able to allow us to diagnose tumors early, but even at the time of diagnosis, know exactly what the mutations are. That's the exciting part of it. So we know what drugs to give them very early in the disease setting. So it's because of setting up the precision medicine that we were able to get this. Now the next goal was to set up institutional biobanking. So historically, a grateful patient gives a surgeon or a pathologist a gift, usually the surgeon. They never meet their pathologists. And that gift is used to set up a bank, and they store tissue from excess tissue from the surgery, and then try to use information from that surgical material. But the problem is to try to understand the disease. But the problem is there are a lot of things that we could do with that surgical material that's far beyond what the surgeon would have thought of doing 5 years ago. We can take small amounts of that tissue and grow it into what we call an organoid. And that organoid will be the tumor growing microscopically. Actually, not so microscopically. You can get these organoids up to half a centimeter or a centimeter or so. What's more, and you'll hear much more about this from Kristy, we can implant that subcutaneously in a mouse, a severe immune-compromised mouse, and the tumor will grow there. So we can watch that tumor grow in a true in vivo environment. This is not a cell line. This is the true tumor of the patient you're trying to treat. Is now either growing in a dish or it's growing in a mouse. And we can then treat that dish or the mouse with drugs and see what's going to work for that patient. And then sometimes in real time, we really figure out the drug combinations that will work before we make a therapeutic decision of whether the patient should get that drug. But that's what the future is. One of these organoids, and one of the most exciting ones, is work done by Howard Fine, a brain cancer doctor just down the hall from me. And he actually takes skin biopsies from his patients, de-differentiates them into stem cells, and then puts them in a broth that allows them to grow into a brain. This is very futuristic. If you're interested, I can send you the CNN link, because he was interviewed on CNN about this, because I'm the only person in the world who could do it. Now many people can do it, but because he's shown people how to do it. But the beauty of this, these brains look like very early brains. They're not yet vascularized, so they don't get much bigger than half a centimeter or so. But I walk into his lab, and his technician pulls out a flask, swirls it around, and there are like 200 little brains floating in there from a single patient. He now puts each of those in a different little well with the same broth, and then takes out their tumor, puts a fluorescent protein in it, and then implants that in the brain. And the tumors grow in the brain. And it looks exactly like the tumor growing in the patient's own brain. So he's reconstituted that glioblastoma into the patient's own brain growing in a test tube. And then, once again, let's throw drugs at it. Drug combinations, what's going to work? So this is very, if you told me this 3 years ago, I would not have believed it, but it's now happening. By the way, you can also grow Alzheimer's brains and Parkinson's brains, and et cetera, et cetera. So we're working together both in neurobiology as well as in cancer to make this work. Now, to get biobanking to work properly, and you want to do things like this, first of all, you've got to get patient consent. So I have to say my biggest struggle when I arrived was, how do we get a universal consent form so that patients who are going to have these surgical materials will agree that we can do research on them and try to learn something. And that means you have to deal with the lawyers. But it happened. It took us about 2 years, but we now have patient consent form that all the lawyers at the hospital and the college have signed off on. And so all these tissues we're collecting and these organoids we're growing and the patient-derived explants, these are all, we now have freedom to work on these in a de-identified way. You can't get back and find out whose brain that was, but you can actually link it to the patient's information, like what was the pathology report, did they respond to drug X, Y, or Z. So the researcher comes in and says, I want to look at 50 triple-negative breast cancers. I want to know whether it was metastatic and what organ, and I want to know whether they responded to drug X, Y, Z. And so you go into the database, and if we've properly annotated it, as we plan to do, you can get that information. And what's more, you can get the sequence of the genes in all 50 of those tumors and see if you can correlate the response to therapy with the mutational events. And we should be doing this on every single patient in order to advance so that we can share information. And if one person responds dramatically, in particular mutational background, to a given drug therapy, and someone else comes along with the same mutational background, they should get the same drug. But we don't share it today. We don't share it because none of this information is publicly available. So by de-identifying it and get the patient's consent, we can share the information and learn from each other. And we're trying to work with the other institutions in New York to do that. So that's what this universal consent form is all about, making maximum use of the surgical material biopsies we get from our patients and being able to use this also to test drugs. There's also something called epigenetics. How many in the room have heard of epigenetics? Ah, well, that's amazing. 7 or 8, 9. Maybe the New Yorker, anybody maybe read the Sid Mukherjee's article in the New Yorker about epigenetics. Anyway, epigenetics is not the sequence of the DNA, but how the DNA gets modified differently in different tissues. And even this modification can be affected by what you eat or stress in your life, et cetera, et cetera. And it turns out that cancers also change their epigenetic signature. And so that turns out to be a really important area to study. And again, Kristy is looking at drugs that will alter the epigenetics of the tumor as well. And some of these are looking really exciting in clinical trials. In any event, all that information is searchable and connecting with the tissue, and you can now implant it, test it, come up with an idea, and use this biobanking to make this all happen fast. So experimental therapeutics program is really what we're going to talk about today, and that's, as I say, Kristy will really get into this very deeply. She is the leader of this along with Giorgio Ingriami at Weill Cornell. Giorgio is one of the world's leaders in figuring out ways to get human tumors to grow in mice. I'm not talking about cell lines, I'm talking about the actual tumor coming straight out of the patient, going straight into the mouth without ever growing on plastic and thriving there. There are lots of tricks to do this. But once we have the biobanking set up and we have all the permits, then we can take the tissue, if it's properly preserved, which means flash frozen, straight out of the bank and put it into the mouse and get it to grow. And so this is, I'll let Kristy tell you more about it, what she's doing here, but it turns out to be much more economical for us to expand those mice and have all those tumorous patient-derived explants growing here than to try to do it down in New York where space is very expensive. And in addition, having it here means that all the technologies that are being developed here at Cornell, imaging, et cetera, are available to us to interrogate in great detail these tumors as they're growing. And we're working with the bioengineering school as well, and I'll get on to tell you a little bit more about that in a second. So that's the excitement. Now the problem is, if you want to test a bunch of drugs, either in these organoids or in the patient-derived explants, you actually have to get the drug. And a lot of the drugs that are most exciting are the ones that are not yet approved. So Kristy will get, she'll get a clinical trial with a pharmaceutical company that has a very exciting new drug coming into the clinic. It's not yet commercially available. It's not yet approved, but she's seeing patients respond. Their tumors are like going away. Lymphomas having a dramatic response, and she wants to know, can I test this in my organoids or in my patient-derived explants and figure out how to add a second drug to it that'll make it work even better? And so typically, historically, you would then go to the pharma company and say, can I have some of this to test in my mice? And about a year and a half later, after the lawyers our lawyers took to their lawyers, and back and forth, they'll say, we'll give you a little bit of it as long as you don't use it in any model except that one. And you cannot possibly ever add another drug to it. Okay? You sign your life away. And that means after a year and a half, you get a little bit to do one experiment. That's not worth it. So what we do is we go to China, and we have the compound synthesized, because by the time it's in the clinical trials phase 2, the structure is actually published. You have to go to the patent, find the structure. We send it to our collaborators in China, whom I've formed a relationship with. Because we do this on a bulk way, they give us about a 90% discount in cost of synthesis, and they give us the drug. We validate that it's truly the same drug that's going into our human patients, and that's what we're putting into these patient-derived explants. We're now fully free to combine those with any other drug. In theory, we're violating a patent here, and because it says that nobody can use these commercially, but we're not actually making any profit from it, so it's not. I would love for a company to sue me for doing this, because we already have several clinical trials that have been initiated because of us doing this that would have never happened. And I'd like to see it on the front page of the New York Times that a pharma company sues us because we figured out how to cure cancer with their drug, which is, by the way, making them a huge profit. So we're not worried about that. So that's our preclinical pharmacy, and that gives us the opportunity to test all kinds of combinations in these organoids, the brain-implanted glioblastomas, the lymphomas that Kristy will talk about. So basically, this little picture shows you what we do. The patient's tumor is biopsied or surgically removed. We do detailed sequencing, in some cases, as I said, full exome, RNA-seq. We bank it in a way that can also be used to grow these organoids. And then we decide, once we test drugs in the organoid, which ones we'd also like to test in an in vivo setting, because it's a lot more expensive to do the mice than to do the organoids. And that gives us some confidence it's worth trying the mice. By the way, turns out if you add nitric acid and sulfuric acid in combination to any organoid, you can kill it. That's a great drug combination. Well, it turns out you can kill the mice with that combination too. So that's one of the reasons we have to go back to the mice, because we want to know whether the drugs are tolerated at the dose that kill the tumor. Does it not kill the mouse? And then we hope if it doesn't kill the mouse, it won't kill the dogs or the humans. But that, you see what I'm talking about. Sometimes drug combinations can be lethal. So that's why we have to do this. This is my final slide. We have a number of joint initiatives between Ithaca and Weill Cornell. And I'll just tell you a few highlights here of what's going on. We just a few weeks ago announced that we were awarded a National Cancer Institute's grant, multi-investigator. This also involves collaborations with MD Anderson, Sloan Kettering, and UCSF. And this center will take advantage of the technologies being developed at the bioengineering school. And the PI of the grant is Claudia Fischbach here in bioengineering in Ithaca. I'm the co-PI. And what we're doing in this case is we're taking these little organoids that we can uniquely grow from the patients and putting them in devices that the engineering school is building, like microfluidic devices. And we can then use the special optics that they have, as well as flow systems and metabolomics, mass spec metabolomics, to look at everything that's happening in that tumor in a controlled environment. So we can watch what it's eating, see what it's spitting out, watch how it's moving, and then follow, even in the live cells, changes in individual metabolites, glucose consumption and products, et cetera. And this is giving us a detailed understanding of how the cancers are growing and how they're invading. So we're very excited to having gotten that grant. So we also are, I'm on a task force that's involving people from Ithaca and from Weill Cornell to try to figure out how we can more integrate genomics. As I told you, we're undergoing a revolution now in genomics. By the way, it costs $13 billion to sequence the first human genome. $13 billion in about 10 years. Today, we can do a human genome for $5,000 in 1 day. So you can see why this is changing. If you told people you could do that even 10 years ago, nobody would have believed it. But the technologies have just advanced so quickly, and it's going to even get better and easier to do and cheaper. So we have to take advantage of all the information we can get from this. And then the problem is, how do we develop our educational system here to train the next generation to learn about these technologies and what they can understand from genetics and epigenetics. The advantage here, of course, is that there are many model organisms being studied here in Ithaca, from fruit flies to dogs, and we can understand, in many cases, much faster in these relatively inbred species, things that would take forever to tease out from the mixed American populations. That's how genetics works. The more inbred, the easier it is to figure out the genes that are a problem. And then finally, I just want to mention another gift that was just recently announced last week from Ellen and Gary Davis. And this gift is to support an immune therapy core. I mentioned earlier that there's a lot of excitement around immune therapy, and that precision medicine is also trying to figure out what's going on in the immune microenvironment of the tumor. So in order to do immune therapy trials, we need to get as much information as possible about what's going on in the immune system in that tumor. And very often we see that there are what we call killer T cells sitting there at the edge of the tumor trying to get in, like particularly in pancreatic cancer. But so they know they should be going in and killing the cell, but there's a message going out from the tumor saying, no, no, don't kill me. They have a cloaking device, you know, they can like hide in there. So it's, the immune cells want to attack, but they can't figure out how to get in. And so we can actually pick that up from these various technologies that are developing, from the sequencing technology, the imaging technologies. And the problem is to figure out how to break those barriers, you know, eliminate that cloaking device, eliminate the brakes on the T cells that otherwise allow them to attack. Sometimes even the innate immune system will attack these tumors. You know, there's a lot to be learned here, but we know this is working. You know, now 40% of melanomas in the metastatic setting are cured, or at least, you know, we're looking now at 5-10 years without relapse. That was unheard of 5 years ago. 20% of breast cancer is showing dramatic responses. Roughly 20% of lung cancer. So this is something, but right now we can't really explain why this person responds and this person doesn't. We have to have this immune core to figure it out. So a very nice $2 million gift from Ellen and Gary have allowed us to set this up. And we'll have, this again will involve collaborations between Cornell, Ithaca, and Weill Cornell. So I'll stop there and let Kristy take over.
Thank you. Yeah, I have different sides. That would be great. Just gonna hold on to this.
Is this right?
That's it?
Okay.
Yep. So Dr. Cantley just gave you a good overview of our whole, I would say, not even just cancer, but our whole endeavor that's sort of happening across campus. I'm going to focus down on one particular aspect of it, which is our experimental therapeutic development process, which we've named PATH, and tell you a little bit more about how we're using that. So just to set up the problem for you, Dr. Cantley talked about this a little bit. When you are making new drugs, it takes a long time, as he told you, 16 years, something like that. But it's also hugely expensive, and one of the reasons it's hugely expensive is because we're pretty bad at it right now. We haven't gotten to that place where we can pair up all that genomic information that we're getting and epigenetic information we're getting with what the right medicine is. So we have to do it empirically. So we go from thousands of possible drug targets. I can maybe point. We go from thousands of possible drug targets that we start with some sort of high throughput screening, maybe in cells grown in a dish. We get some more promising things. We go maybe take it into mice. And then only the very best things that look the most promising go into humans. But look here, it's showing 5 compounds here that go into humans. It's still only one of them makes it to FDA approval. And in oncology, that's even much worse. It's more like 1 in 15. So at the end of the day, what that means is the cost of making a new drug is estimated to be $2.6 billion. That's one new drug that gets FDA approved. Obviously, that's not sustainable because we know from precision medicine, this person needs this drug, this person needs this drug. We're going to need hundreds of drugs. And we can't wait 20 years for each one of them to be developed. So we need to get faster and we need to get cheaper in our drug development. So that's why we created PATH. And like I said, that's our experimental therapeutics development. So this whole process of making new drugs, we try to think of ways all along. We basically tipped that last slide on its side, and all along this pyramid of development, getting progressively more specific and closer to human clinical trials, we tried to fix it so that it worked better, so that the things that actually make it into the clinical trials work and get approved. If you think about it, if you can even just increase it by 2-fold, you'll suddenly save a huge amount of money and time. So one way we've done that, and you've heard a little bit from Dr. Cantley about some of these things, and you're going to hear some more in a minute from some of my students actually using these in real life, are with organoids, so tissue grown. Instead of cells grown in a dish, it's actual 3-dimensional tissue grown in a dish, and you can do high throughput screening the same way, but the things are more accurate because they have the microenvironment there and they're growing in a much more realistic situation. Brains even, you know. Not in my world. In lymphoma, we don't do brains, but we do lymph nodes on a chip. And then also those mouse models that I was talking about that we used to use, we try to figure out ways to make those better. So one way to make them better is to implant the human tissue right into an immunocompromised mouse. Dr. Cantley was talking about that too. So we can take tissue out of a cancer patient, implant it into an immunocompromised mouse, and just grow it there. That lets us test the tissue from the actual patient. Also lets us potentially make several different mice carrying that same tumor. You can think of them like patient avatars. Right? So you have your tumor growing. You can test multiple different drugs or drug combinations. You can see which one works the best in that mouse. And then you go back to the patient, potentially, put that combination in the patient.
Does that actually go on today? Or like
So,
clinically do that? Or
so I would say we're not at the space yet. We're getting close, but we're not at the space yet where we can reliably get the tumors to engraft 100% of the time. For some, engraft into the mouse. So we can't promise it to patients as we can do this every time, but we're getting close enough with some types of tumors that the take rate is high enough that we can start, and we are starting to think about doing clinical trials where the standard of care for the first line of therapy is usually set, right? So you wouldn't change that, but what you could do is take the tissue out before you treat, grow it in the mice, and while the patient's getting their first line of therapy, test the different drugs. Then if the patient relapses, you're ready with the next best possible thing. As an oncologist now, I'm usually shooting in the dark. And when a patient relapses with their lymphoma, I pick, a regimen. There's like 3 or 4 possible ones. If that doesn't work, I do another one. If that doesn't work, I do another one. And I don't have anything to guide me to tell me which one. So it would be nice to have either some genomic information or some PDX information to help me guide in that process. And that's sort of what precision medicine is all about.
Dr. Richard,
Yes?
the mouse brain tumors,
Uh-huh.
Are they studied locally at the site of grafting, or do they behave systemically, and can you study the whole mouse?
So we've seen that too. So some tumors are more metastatic than others. And we've actually seen that even in our lymphoma organoids, which you don't think of as a metastatic disease because it's kind of spread around all through the lymphatic system from the get-go. But even in our PDX mice, we see some tumors that like to travel from their original site and other ones that kind of more stay put. So that's...
Do they travel as they would in the human patient?
Well, we implant them under the flank. So that's kind of an unnatural place for them to grow in the first place. So I would say their tendency to migrate, which you're going to hear more about in a minute, is potentially different between different tumors, and we can study that, but I can't say that it's exactly perfectly a replica. You can implant in the PDXs, not just under the flank, but you can implant, for instance, a breast cancer in the mammary fat pad and watch it metastasize to its places. So it's possible to do that. The other thing we have is our typical mouse models. We change some genetic lesion and we make a mouse. We've now figured out a way, oh, and I should say, I've been forgetting to give the proper credit. Our organoids are done in collaboration with Ankur Singh here in the engineering school. He's one of the people that's developed these lymphoma organoids. And our PDX mice are done with Giorgio Ingrami down at Weill, but also with Bob Weiss up here at Cornell. And we have similar facilities and imaging machines so we can look at the tumors both places and compare results. The genetically engineered mice, we have taken advantage of a technology you might have heard of called CRISPR, which is starting to revolutionize how we can edit genomes and make very specific changes in the genomes. And John Scamenti here is doing that, one of the experts in that kind of work. And so we're making more reliable, we can make the exact base pair, the exact specific change in the DNA that exists in a human lymphoma, for instance, in a mouse, and see what that looks like. And then the last thing is something that my lab works on a lot is clinical trials in our veterinary patients. So mouse models are great. You can make very specific lesions, you can do all kinds of things, but those are never spontaneous, well, very rarely are those spontaneously occurring tumors. Spontaneously occurring tumors are a little more similar to what happens in humans. Patient gets a tumor, right? So the dogs that come into the veterinary clinic with lymphoma, they are sharing our environment, they're drinking our water, rolling in our pesticide-covered lawns, doing all the things that we do, and they get lymphomas just that look just the same as ours. So we can do clinical trials with these new drugs in the dogs and see what happens. And then hopefully when we get to the top of the pyramid of the clinical trials, we are more accurate and getting our drugs approved faster. So in order to demonstrate to you how this PATH program is working, I'm going to have two of my students talk to you about what they're doing with the PATH program. And I have to give you a little bit of information about lymphoma in dogs, which I've told you some of. It's very similar to non-Hodgkin lymphoma in humans. It's one of the most common cancers in dogs, so we see it a lot. There's up to 250,000 new cases in the US every year. And the median survival time with treatment is only about a year. So the whole disease process happens faster than in humans. We can do these trials faster and these studies faster in the dogs. And the 2-year survival rate, unfortunately, is 20%. So the dogs need some help too. We cure about 50% of humans with lymphoma, but the dogs don't do as well, so they could definitely benefit from our research. So I'm going to have two of my graduate students come up. The first one is Dr. Wilfred Lung. He's a veterinarian, and he's a Ph.D. student, so he's studying to get his Ph.D. And then after him will be Dania Villarnovo, who is another DVM, who is also studying to get her Ph.D. And they're going to just tell you how they've used this PATH system to learn more about how to treat lymphoma. Okay, we'll just clip that onto you here. You can put that in your pocket if you want.
Sure. All right, so I'm interested in studying how migration happens in lymphoma. So essentially how cancer cells spread throughout the body. Now this is important because this clinical feature is highly prognostic. So a patient with stage 1 disease where the cancer is located within just one lymph node does a lot better than someone in stage 4 where the cancer has spread throughout their whole body. And so how cancer spreads is through a combination of cues and signals which tells these cancer cells to migrate. However, the exact mechanisms and drivers behind this are largely unknown in lymphoma. One of the things we're looking at is testing to see whether the WinT pathway is on lymphoma. So essentially what this does is it's a signaling pathway that's evolutionarily conserved, and it's only turned on during development. However, it's co-opted by a lot of different cancers, such as colon cancer and breast cancer, and this results in cancer cells or sending signals to these cancer cells to survive and spread throughout the body. In lymphoma, the WinT signaling pathway is unknown whether it's turned on or not. So in order to test this, we look to see whether a critical effector protein called BetaCatenin is present in these lymphomas. So what you can see here is that a black band indicates that the protein BetaCatenin is turned on. And as you can see through a various array of different lymphoma cell lines that we see different levels of BetaCatenin present. And so the next question to ask is whether inhibiting this pathway would result in decreased cell growth or inhibition of the cell's ability to migrate. So in order to do this, together with our collaborator, as Kristy mentioned, down in biomedical engineering, we've made this three-dimensional structure called an organoid. So essentially what this does is that we encapsulate these cancer cells within a three-dimensional structure, and our collaborator has shown that it closely mimics what you see in the native tissue. So we're able to stimulate what these cancer cells are encountering inside the body. And you can see here, this is this three-dimensional spheroid structure that we have the cells inside, and we can also incorporate different peptides and ligands that the cells would naturally encounter inside the body. And we think that this system would allow us, would bring novel things which we can't see if we just grow these cells in a culture media. And when we grow these cells within an organoid and we stimulate them, as seen on the right here with a WinT ligand, we see the cells actually cluster together, which is quite unusual. And what we think what might be happening is that either the cells are proliferating really fast or they have an enhanced migration potential. So we're looking into that. And we're seeing whether inhibition of the WinT pathway would reverse this effect. Furthermore, we've tested different drug combinations in this organoid system. And so you can see here a different group, the cells treated with increasing drug concentrations, with the orange bars indicating cells grown in a culture media and the blue graphs indicating cells grown in organoid. And so you can see by this that the cells respond differently within the organoid group and cells grown in culture. And so we think that moving forward, that this system would give us benefits to just studying cells within a liquid media that we wouldn't see otherwise.
Okay, so this brings me to my project. So I've been studying a protein called Fascin-1, and this is a protein that's involved in giving cells their structure. And it's actually found in really motile cells, so cells that are able to travel from one part of the body to another normally. So this protein has been found in really high levels in cancerous tissues in comparison to their normal tissue counterparts. So the idea out there is that perhaps this protein is able to give these tumor cells the same kind of motility and able to move again from one part of the body, so from a primary tumor site to other sites, in other words, to metastasize. So we've been collaborating with an investigator at Weill Cornell, and he had developed a drug called G2. It's a small molecule inhibitor to this protein fascin-1. And Dr. Huang has actually already tested this drug in his own laboratory using breast cancer cells from humans that have been injected into mice. And what he has found so far, as shown in this image, is that when this drug is injected into mice that already have these human breast cancer cells, they actually have a lot less metastasis to their lungs. And this is indicated by the smaller areas of blue, red, and green. And this is also in comparison to these control mice that did not receive any of the drug. So this drug appears to be effective, at least in these mice, to prevent metastasis of these breast cancer cells to their lungs. So in our laboratory, we wanted to test, first of all, well, can we find this protein in patient samples that come from dogs that have lymphoma? So these are actual patient samples that are submitted to the veterinary school here at Cornell for their initial diagnosis of lymphoma. And once they're diagnosed, we can then acquire the tissues and do different tests on them. And so I obtained these histology samples, and then I stained them, and this is indicated by all the brown that you see here. I stained them to see where exactly I could find this protein fascin-1. And when we take a closer look at this tissue, as shown in this bottom image here, we can see that in this particular patient, there was a lot of expression of this protein fascin-1. And this was actually very typical of a lot of the patients that we saw. The next step was then to test in a similar fashion whether we could see fascin-1 expression in cell lines. So these are cells that we obtained a long time ago, actually other researchers did, from patient samples, and then they've been grown in laboratories and propagated for many, many generations. So we obtained these cells and then tested them again for this fascin protein. And what you can see is that it really depended on which cell line you looked at. So in this cell line, CLBL1, this is originally from a B-cell lymphoma from a dog patient. And you can see that, again, by this brown staining, there were a lot of cells that had expression of this protein. And this is in comparison to other cell lines that didn't have it in very many cells, like in 1771, or didn't have it really at all expressed. So it really did depend a lot on which cell line we looked at. The next step was then to see, well, can we obtain this drug from our collaborator, G2, and see if we can test it with these same cell lines and see if we can then use it potentially to treat lymphoma. So these cell lines that I just showed you, the canine lines, but we also used human cell lines, and this is just an example. These results were very similar, regardless of what cell line we looked at, both in dogs and in humans. And what we did was we treated the cells with different concentrations of this fascin inhibitor, and then we did it for different time points. And then we counted how many live and how many dead cells we had. And what you can see by this darker blue line and the yellow line, so this is the higher concentrations of the drug, you can see that the number of live cells decreased dramatically over time. And those cells that did not receive any of the treatment, so this is the lighter blue line shown here, actually kept proliferating, and their numbers actually increased during this time. So one possible reason why they decrease is potentially we killed them. And so this graph here shows that indeed that's at least part of the reason why we have a decrease in the cell concentration after treatment. So again, the yellow and the darker blue lines are those that received the higher doses. And you can see that the number of dead cells increased over time, whereas those that did not receive any treatment did not really die as much. So this indicated that indeed we could use, at least in the lab, this drug to kill lymphoma cells, and then also to potentially decrease the proliferation of those cells in culture. So then this leads us to our future trials. So for both this fascin inhibitor G2 and also for the WinT inhibitors that were spoken about previously, we can then test this, take this one step further from our lab to PDX mice that Kristy just spoke about. So these again are mice that are injected with tumor tissues from actual dog patients that have lymphoma, and then they grow a similar kind of tumor in their bodies that we can then use to study further. So these same mice, we are planning on administering this drug and then seeing, well, can we kill these tumor cells or at least inhibit their migration or metastasis to other sites. And then the other step will also be to actually test it in actual canine patient samples or patients that come to the veterinary school here seeking treatment for their lymphoma.
All right, so that was 2 quick examples of how we're using the PATH program to develop new drugs for lymphoma. And I showed you a pyramid before, but I think probably it's a little more accurate to say that the PATH program is more like an engine where all the different pieces are constantly, iteratively feeding back to each other to make things better and to get a more accurate and quicker path, to have a pun, to FDA approval. And then lastly, I'd like to just, and I wanted to mention one more thing. Along the lines of projects that our PATH program has enabled, we just recently received a $5 million 5-year grant from the Leukemia and Lymphoma Society that centers around the PATH program, and it's a cross-campus effort. Part of it is, the majority of it is actually down at Weill, Cornell, and part of the project is done up here in Ithaca. So it's a true cross-campus project. And we also just used the PATH program as part of our CTSA renewal, and that's CTSA is the Clinical Translational Science Award. It's usually exists at medical schools, and it helps bench-to-bedside research. So the PATH program has definitely been instrumental in helping us to get grant funding. And it's a true cross-campus effort. You can see all the researchers here. I only listed the principal investigators. Not all of their research groups. It would be too much for the slide. But you can see a nice split between New York City and Ithaca, which is a really great thing, I think, for the project. And that's all I had to say.
Okay, thank you very much. And so we have time for a few questions for any of our speakers, so if you'd just like raise your hand, and Kristy, you can field the questions and help direct them. So.
Yeah. Fire away. You asked some while I was talking.
So,
Yeah.
Just a quick one. You showed the traditional drug development timeline. I mean, how much is this going to shrink?
Well, that's kind of why I showed this.
Sounds like the research is still going on.
Yeah. So, it's sort of it's an evolution. I think it's like Lou was saying. If you said 5 years ago, how long would it take? I would have given you a different estimate that I would give you now. And I think this is the key to how we think about it. We used to think of it as a very linear thing. We're starting to jump ahead with the dogs, for instance. Okay, so let me give you an example. In humans, you are not allowed to give a drug in combination until you've tested each drug by itself, right? And so just that initial test takes a few years. So if you have to do each one by itself, what if you need a combination of 3 drugs to get a tumor, right? In dogs, we are not constrained by that. If it is not toxic, provided we can show it's not toxic, and we can do that with research dogs very quickly, we could jump ahead and put together any combination we think is great based on our preclinical models and try it out in the dogs. And then I think, hopefully, it becomes one test instead of several sequential tests. So I think this can speed up the process, but we haven't. Ask me in 5 years. Yeah, so I think there's definitely ways we can speed up the process with this, but we'll have to look in retrospect and see how well we did in another few years.
All right, we'll be back next year.
question over here.
Yep.
What's your experience with the FDA and the legislation? And do you feel that that is a big
It's a little bit of a challenge in the dog studies because not from the FDA so much as people's perception of the FDA. So the pharmaceutical companies are very gun shy about doing trials in dogs because they're worried that something is going to happen in the dogs that wouldn't happen in people, and that will kill their drug. So we've actually gotten the FDA at an Institute of Medicine workshop to say, no, that isn't true. But even with the FDA's assurance, I still am having a little bit of trouble with the drug companies sometimes. So I think it's about establishing relationships and establishing, getting over that gun shyness to do that. Yeah. The other thing I would mention, though, in the dogs that is pretty interesting is for immunotherapies, dogs are a great model for immunotherapies because they have an intact immune system that's been challenged just like our immune system. That's not true for laboratory mice. For dogs, immunotherapies are regulated by the USDA, not the FDA. So there is some potential there for speed that does not exist when you're being, governed by the FDA. Yeah, because their standards are a little bit more lax. They just want an expectation of efficacy to give you conditional approval, which is not what the FDA requires, as you might know. Yeah.
Kristy, we talk about a global economy, but global medicine is another, obviously
Yes.
right there. And so with those forms filled out, the disclosure, are you able to keep a database for people globally treating those types of cancers?
So we could do that, and we have done that a little bit. I have a collaborator actually in Malawi who treats human lymphomas. And he's actually sending us some Burkitt lymphomas for a researcher at Weill Cornell to sequence to help compare. But again, it's a little bit like the dogs. Not that I'm comparing people in other countries to dogs, but in other populations, we don't know if their tumors are molecularly the same as the ones we've studied here already. So we sort of have to do a little groundwork before we can do that. But yes, I think it's a good idea because anytime you can compare across groups, you can learn things.
I should say that's part of the reason. That's part of the reason we were awarded that precision medicine grant, the $20 million, is because our catchment area in Queens and Lower Manhattan and Brooklyn has a lot of immigrants from Asia, and the diseases they get. In fact, even in lymphoma. Or AML rather, even in AML, leukemia, the mutational events that dominate their disease is different from the mutational events that dominate Caucasians. And we don't know why. Is that ethnic? Is it a germline difference? Or is it lifestyle, food, et cetera?
Yep.
That last sentence was almost answered the question I was going to ask. I was going to say that we know the 80% of the genetic abnormalities of the tumor.
Well, this one Lou definitely has to answer.
And if the answer is yes, then do we understand that as well in the veterinary portion?
Yeah, I'll give this back to Kristy too, but I think, we certainly know that lifestyle can dramatically affect cancer, lung cancer being the first dramatic one. Actually, before that, chimney sweeps getting testicular cancer was known 200 years ago as a lifestyle effect. We now know it's carcinogens and smoke that caused the testicular cancer because the little boys went down the chimneys. So we knew it for a long time. But today, the biggest risk, now that tobacco is becoming a little more in control, is insulin resistance. I mentioned that the pathway I work on is both important in insulin and also in cancer. And that pathway in insulin-resistant patients is hyperactivated, and it's growing evidence that we're going to see a large increase in cancers, particularly breast, endometrial, colorectal, pancreatic, all of which use that pathway as a consequence of the obesity and insulin resistance that we're seeing massively occurring in the United States. So that is going to dominate.
You just studied cats.
Yeah.
Cats, yeah.
That's good.
Yeah, I think we have to pay attention to what we feed our animals as well.
Yeah, there are obese animals. So again, you know, the animals share our environment, so they have the same issues that we do. There are obese dogs and cats, so and they eat the same foods. I didn't mention that lymphoma incidence is rising in both dogs and people. It's nearly doubled since 1970 in people. In dogs, we know it's going up and they're getting it at a younger age, but we don't know why in either species. So potentially there's an environmental thing there that we can find that's a link. Yep.
All right, thank you very much.
Yep.